Okay. Good morning, everyone, and welcome to this first session of Thesis presentation. My name is Martina Coppari. I am a researcher and teacher at the Polytechnic University of Marche in Ancona, in Italy, and I will be the moderator for this session. Together with me, there is the jury composed by Ana Maria Bafediau, which is a postdoc and teacher in the Ghent University, and Nagore Gonzalez Soto, which is postdoc and teacher in this same university, and Gautier Char, which is an associate professor from Brest University. So before we start, I just want to remind the students that you have 15 minutes for your presentation, followed by 15 minutes of comment and discussion with the jury. And when there will be 2 minutes left for your presentation, I will stand up so that you know that you need to go to the conclusion. Okay, so we can start. I invite the first student, Evelina Zinkiewicz. She's going to present the thesis titled A Genomic Guided Approach to Natural Product Biosynthesis Materials Sustained with the Coral Leptococcus armentosa.
Everybody can see you.
How do I switch this one?
With this one. Or you can choose to talk louder. Or you can take the microphone. Up to you.
Hello? Can I start?
Yep.
Good morning, everyone. My name is Evelina, and I'm really excited to talk to you today about my master's thesis project, which is called A Genomics Guided Approach to Natural Product Biosynthesis in Bacteria Associated with the Coral Leptococcus armentosa. And the main question driving this work was basically what bacteria can we find on this coral, what chemistry do they produce, and how much of it have we never seen before. So to understand why that matters, I'm going to start with 3 fun facts. First is that sponges, tunicates, and corals are one of the most chemically rich sources of new therapeutics. So for example, eribulin and terbinafine are both cancer therapeutics in clinical use today. And pseudopterosins, which are very well studied for their anti-inflammatory properties, have all been isolated from marine invertebrates. Second is that many natural products historically credited to the host animal are now understood to be made by their bacterial symbionts. So that's why there's a growing trend in studying the bacterium of invertebrates. And third is that bacterial genomes hold more chemistry than they ever express under standard lab conditions. So this is what is driving the genomic approach. And this is called cryptic biosynthesis. So our study organism was Leptococcus armentosa, a common Gorgonian octocoral, also called a sea fan of the Northeast Atlantic and Mediterranean, found anywhere from 10 to 200 meters depth, and it forms dense animal forests, increases habitat complexity, and supports biodiversity, which has a huge environmental role, and yet very little is known about its microbiome. So we had 3 objectives for this study. First was to build a genomic resource through whole genome sequencing, assembly, and quality assessment of the bacterial collection. Second was to characterize taxonomic novelty, so to place these isolates phylogenomically and identify which ones are potentially new or undescribed. And third was to map the biosynthetic repertoire, so to detect the biosynthetic gene clusters, group them into families, and prioritize candidates for future natural product discovery. So we had 8 coral colonies sampled by scuba diving at around 12 meters depth in Arabida Natural Park in Portugal in March and June. And we had 2 cultivation media, marine agar diluted, and marine color extract, which is basically the specific marine medium specifically made by our team to mimic the coral for the bacteria to grow. And we had 3 different oxygen conditions: aerobic, microaerophilic, and anaerobic incubation at 18 degrees Celsius to favor slow growing in oxygen diverse taxa. So this is pretty much what it looked like. All of these pictures are the genus Aquimarina because it is a very well-known bioactive compound producer. In any marine microbiology lab, Aquimarina is the legendary genus. So we had 8 coral colonies sampled. 328 isolates were picked, purified, and identified. And from then on, we got 94 genomes were sequenced, sent to us in FASTA files. So this is where my work has started. all of the stuff that I talked about before then was done by a PhD student before me. So I have started here. It would be great if I could show you each one, but I can't. So first we had a genome assembly. All of our genomes were sequenced by Illumina, and then Aquamarina was sequenced by Nanopore on top of that. And all of this work was done in Galaxy, which is a software. And assembly was done with Spades, and quality control was done with Checkam 1 and Checkam 2. Then the taxonomy was assigned in Genome Taxonomy Database to the tool GTBTK. And then the biosynthetic potential was detected through anti-smash, which is also a software. And similarity networking was done in BigScape 2, which paired the biosynthetic gene clusters into families, and visualization was done in Cytoscape. So, to our results. We have found 94 genomes spanning 50 genera and 6 different phyla. 33 of our 51 genus level lineages were represented by one single genome. 6 lineages were undescribed in any reference database, and 3 had no culture representative anywhere in the reference database. So the main insight from this figure, this beautiful massive phylogenetic tree, is that the microbiome of the squirrel is very diverse. So to assign taxonomic novelty, we used the criteria taken from the paper by Jane. A strain is considered novel if its average nucleotide identity is less than 95% and its alignment fraction is bigger than 0.5. So applying these criteria to all of our genomes, we found that 76% of our 94 genomes are potentially novel species or have no GTTB match. And one strain specifically was assigned to a family, Sepiaceae, that is outside any described genus. So it's not only a potentially new species, but a whole new genus. So to visualize our statistics on the bar chart, we can see that from our 94 genomes, 4 were known species, 22 were potentially novel, 19 were inconclusive, and 49 had no GTTB reference. So first, now I'm going to talk about biosynthetic potential, and first, what is a biosynthetic gene cluster? It is a group of physically neighboring genes that together encode the set of enzymes to produce one natural product. So for example, this is our strain Pseudoalteromonas, which produces violacane, which is an antifouling, antimicrobial pigment. So we have a bunch of genes with a regulatory sequence right there, and they together produce violacane. That's it. So, what have we found? We have found 615 BTCs were detected across our 94 genomes, which is a lot. Out of those, 164 were RIPs, ribosomally translated peptides, 79 NRPs, non-ribosomally synthetases, non-ribosomal synthetases, 78 terpenes, 49 polyketides, and 32 cytosine, and 200 others, which are basically the hybrids of all of these different BTC types. And this is exactly what you can see on this figure right here. We have all of our genomes here, and all of the types of BTCs that they produce here. So we can see that Pseudoalteromonas there on top has the most BTCs. Shewanella has the second most, and that is also very curious to see, because Shewanella is very notorious for having tiniest genomes. And this bar chart represents the percentage of complete BTCs, so whether the BTC was found on the edge of the contact or in the middle. So then we took these 615 biosynthetic gene clusters and we blasted them against a database that tells you which ones produce what, basically. And we have found that 74% of these BTCs return no significant match, so all of that is new to us. And 9% return a high confidence hit to something that has already been characterized. So the biosynthetic repertoire is largely distinct from anything previously described in chemistry. And these on the left are a few examples of the compounds that we found. So we had iron scavenging cytorophores, broad spectrum antimicrobials, antifouling pigments, osmoprotectants, antibiotics, biosurfactants, a lot of cool stuff that can be used in science, pharma, industry, whatever. Then we took those and we grouped them into families. So what is a gene cluster family? It is a group of BTCs across different genomes that are similar enough in structure to encode an identical or similar compound. So we did that in BigScape 2, which scores their similarity based on shared protein domains, domain sequence identity, and whether the domains sit in the same order. And we have two outcomes out of that analysis. It's a GCF member, so it's something that has been connected to another BDC, or we have a singleton. So it's a BDC that has no close relative to anywhere else in the collection. And this is what it looks like. So for example, this cluster is all the same colors, which means it's the same family, especially Acromerina. And the different shapes represent different BDCs. So we can tell that this genus produces different types of BDCs, and the lines are basically how close they are in structure. And if we look at this family, for example, we can see that we have 3 different colors, so 3 different genuses, and 1 shape, so 1 type of BDC. Keep that in mind. So, after doing that, we have found that 30% of them were networked, so connected to other BDCs, and 70% were singletons, so not connected to anything else anywhere else in the data set. Again, huge amount of biosynthetic novelty. And we have prioritized 3 families that were the most interesting. So we had a gene cluster family 45, which is a quorum sensing one. So it's a homoserine lactone system conserved across 4 Azobacteriaceae family genera, which is likely a family-wide trait, possibly shaping coral colonization disease dynamics because it has been possibly selected for during evolution. Then we had a beta-lactose system that had the same architecture across 3 different genuses. So it is very interesting for chemical identification. And we had a terpene cluster, which has been conserved between Rosabium and the unclassified, potentially novel genus. And the next work would have to be done in PRISM 4 and Devreux Predict, which are basically softwares that tell you that predict the chemical structure of the BDC that you're trying to study. So in conclusion, we had 94 genomes from distinct families, which is the first systematic atlas of Leptogorgia sarmentosis cultivable bacterium. 76% of it was novel species, including 1 potentially novel genus, 615 biosynthetic gene clusters with high novelty, so 74% of it was never characterized or seen before, and 3 priority gene cluster families to be studied in the lab with potential applications in a lot of different fields. So what does it mean for us? For the field, it means that we now have a reference atlas for coral microbiomes. We have 94 cultured and sequenced strains available for the community. We have a model for targeted cultivation genomics in understudied hosts. For the society, it means that we have a lot of potential therapeutic leads, as well as microbiome insights relevant to coral disease resistance, and a foundational data set for natural product pipelines. And for the lab, the next steps would be a chemical isolation of any of the gene cluster families that I already talked about, taxonomic description of the new genus, and metatranscriptomics to confirm in situ expression. Thank you for listening. I would love any questions, and I would like to thank my supervisors, Tina Costa and Rodrigo Costa, as well as my co-supervisor, Joana Cosero, that has done all of the cultivation work for this thesis. Happy to clarify.
Okay, we can start with the question. There is one volunteer, otherwise.
I can still stand.
Thank you, Evelina. I think, well, this is as far as my field of expertise as it can be. That's, if I understand well, you're working on the sequencing of the cultivable bacteria.
Yeah.
Do you know what part of the microbiome you're missing by only working on cultivable bacteria?
Yeah, the in situ expression. In situ expression.
Yeah, but you're missing the one that you cannot cultivate.
Yeah, for sure.
Any idea how much of the microbiome you cannot cultivate?
We cannot really say. This was basically the purpose of this work was not to say which bacteria are mostly found on this coral. It was just to find which new ones can we see. There is a big limitation in obviously just sequencing cultivatable bacteria, but the purpose of this was biosynthetic potential out of what we find, and not characterizing the actual like bacteria that are found on this coral point.
And maybe another question: how can you tell from a sequence that you have a cluster of genes and not different or non-related genes?
Yes, so a lot of bioinformatics. So this part is in biosynthetic gene clustering mining. So that is done with anti-smash. It is software with a lot of internal statistics inside how it's done. And it's basically it's all of the BGCs are detected and are blasted against a reference database. So there's a lot of BGCs that we have seen that they repeat in different genomes, but we don't know what it is. So we just assume that it's a biosynthetic gene cluster because it has repeated in many different genomes. It has been conserved through evolution in different bacteria, and it has likely but corals have a lot of bacteria and corals have a lot of developed functions for like quorum sensing, cell-to-cell communication. So if we find one cluster of genes that has repeated and has been conserved through time, we assume that it has something that is there for a reason. So when you do the big SCAPE2, for example, you download a file domain similarity, which is a file, a big reference database of previously communicated potential BGCs, and then you compare your results to that file, and it tells you which ones are potentially BGCs, what they produce. Yeah. Did that answer your question?
Absolutely.
Okay.
Am I allowed this last full question? A question that does not really concern your work, but always wonder, why are sponges, corals, and tunicates so rich in diverse molecules? I mean, they are extremely far apart in the metazoology. Any idea?
I think because they're found in so many different environments, like some are shallow, some are deep, some are tropical temperature, some are in the Arctic. Not in the Arctic, but like there's tunicates in the Arctic, I think. And so they had to adapt through time for different temperatures, different conditions, pressures, and everything. And they had to develop like copybots. They had to develop the adaptive strategies to survive. I think
That's a good explanation.
so. There's a lot of
Trust you.
stuff to be found.
Thank you very much.
Thank you for the nice presentation. I will go for, if you imagine that I'm a chemical industry or pharma that want to give you money to continue with this work. So you thinking this for biosynthesis, so which cluster you will choose if you want to sell me your product going from here?
So this slide is basically the response to this question. We have 3 prioritized families that are the most interesting from terms of I would say if I had to pick one, I would say that one, the beta-lactam system, because I studied the exact architecture of the genomes, and it was exactly the same across 3 different genuses. For example, these 2 are both Vibrio, but this one is a completely random one, unrelated. It has an HMGL condensation architecture, which basically means that it's not just producing, so that would be a normal polyketide that they produce, which is also has properties, biosynthetic properties. But this exactly means that they are making the molecule more complex. So they're borrowing, I think this is secondary metabolism, and they're borrowing specific tools from primary metabolism to create a more structurally complex molecule. That's also really interesting for drugs, for example, because the more structurally complex a molecule is, the more active sites there is for like stuff to attach to. So it's also very would be great to study for pharma, for example. But any of these three would be really interesting to study. This would be a whole novel genus, and that's a bacteria that's connected from a very different side of the phylogenomic tree.
Going further of that, what will be how you will perform that next step?
So I'm not really a chemist, but first it would be doing these softwares. Basically, you put the sequence of the BGC in it, and it predicts the chemical structure of the compound that it may produce. And then it would be to isolate these bacteria, isolate those specific molecules, study them, test them against bioassays. I don't know, and yeah, just study for bioactivity testing. So that will be different bioassays depending on what they do.
I'm going completely the opposite side. Why you started from this coral?
It is very, very common. There are so many of them off the coast of Portugal, and there's barely studies on it done. So it's the abundance of it. So it's really like we only had 8 corals sampled, which are super tiny, and we had 328 isolates picked up from it. So economically, it's very easy to do if we do find something promising. Yeah, so it's the abundance, how understudied they are, and how much potential they have.
Thank you. After my side.
Congratulations for the very nice presentation. Actually, congratulations on your work. It's amazing. It's impressive to see this kind of results also from a massive thesis work. Well done on that. It's not easy to disseminate this kind of research. So yeah, I want to ask a few questions, like more related on the different steps that you follow in your analysis. So, for example, well, starting first from the culture, first. What do you think that the cultural protocol is well established enough that if say we repeat the same way of culturing, we end up possible to end up with the same sequences, of course. But I mean, is it well established to make sure you capture as much as you can capture from your sequencing part, or could be improved, or it could be another culturing protocol that gives totally different results? So it's hard to compare between studies, I guess. What do you think?
So our materials and methods part, cultivation, was focused on discovering as many taxa as possible, because we use 2 different cultivation media. So the one that is mostly used in studies in marine agar, and then our group has also created their own marine medium that specifically mimics the coral environment for the bacteria. And we had 3 different oxygen media that is also not very common to do in cultivation studies in corals. So yeah, and we had 18 degrees Celsius, which is fairly common. But yes, there was a lot of different parts of the study specifically to favor slow growing and oxygen diverse taxa. And if we look at this, for example, there is a lot of information on this picture, but the stars around are the different oxygen conditions. No, those are the culture mediums. And the oxygen conditions are not in this figure. But if someone was to look at this study and the paper that is going to be published based on these results, if they wanted to focus specifically on one bacteria, for example, that has never been cultured before or found anywhere, we can see which oxygen conditions it prefers, which marine medium it prefers. So our study was mostly focused on just covering as many as possible.
Yeah, that's very good. Answer very well. I mean, I was also, I could not see that you compare also between the different cultural media and so on, but this is nice. Okay. Also, then next step, bioinformatics, a huge black box. I mean, you have a lot of different type of analysis that come with the different results. Keep improving with time. You can change analysis and so on. So I was wondering there few things. First is a more general, like within this path that you followed, the different type of analysis. What would you think? Like, let's say you have a new student starting the same work and you have to guide him or her. What would be the step where you would try different things or different methodology? Or would you follow the exactly same steps? Or is there any part, in other words, that you would like to test differently? Because I see there different analysis, for example, the clustering analysis or even the tree, genetic tree, you do different ways. You know, that these things change the final result.
Exactly.
So, where would you put, let's say you have time more work into exploring more different steps from your own experience?
Yes. So all of this is basically tools that are being updated all the time. For example, Anti-Smash was released in 2025. Actually, there's a new version of Anti-Smash. I would use that one instead of the version 7. The Big Scape tool was also released in 2025. I think I'm like the first person in my lab who managed to make it work. So that was tough. But all of these are new things that develop over time very, very quickly as well. So if there was someone in the future doing this, I would obviously suggest like using newer versions and everything. But also I think it depends really on the focus of the study. It depends on what you want to focus on. And then I have no expertise in like the specific bioinformatic things that detect a very niche focus on something, because my study was very broad and like descriptive mostly. So to find as many things, find as much biostudy potential as possible. So I don't think I can really answer that fully because I think it would depend very much on the focus of the study.
Thank you.
Thank you, Irina, and really nice work. I just have one consideration, and do you think that the results that you obtain will change if you sample different specimens from different location, different depth, or from the same location with different depth?
So normally if we were talking about corals in general, yes, obviously, but octocorals are specifically very well known to have a very stable microbiome. So for example, we sampled in June and March. That would be sort of different abiotic factors, but the microbiome was pretty stable and very similar. So yeah, if you're studying octocorals, I would say not really, not significant at least.
Thank you very much.
Thank you.
I think we can move to the next student. I invite
Hello, my name is Laura, and I'm going to present my thesis work that I did at ILVO on eDNA metabarcoding and metagenomic skimming workflows with applications in population level genetic inference with a focus on the common soul. So, ICES, which is a scientific advisory board, has stated over 60% of their stocks are considered data limited. In the Belgian context, less than 2% of the commercial catch is being monitored for biological data. Alongside this, genetic diversity has been acknowledged by the CBD as an essential part of biodiversity, and monitoring targets have been explicitly adopted in 2022. Solea solea is one of the fish species that has data-limited stocks in Europe. It is a benthic flatfish that has large important commercial importance in Europe, particularly for the Belgian fleet, where it represents almost 50% of the fleet's revenue. So, environmental DNA has been proposed as a method to improve data collection for data-limited stocks and for all stocks, really. eDNA is genetic material collected from the environment without directly sampling any of the individuals. It's non-invasive, cost and labor effective. It detects rare and cryptic species. And bottom trawl surveys, which are one of the most common fisheries-independent data collection methods, particularly for benthic fish, have their own limitations such as selectivity bias, they're spatially limited, destructive, and they provide no genetic information. So eDNA has been proposed as a complementary approach to add to these trials to improve data collection. So in eDNA research so far, it's very routinely and common to use it to detect species presence, but expanding that to extracting population level information, so genetic diversity within a species, still remains quite challenging. And some of the limitations to that are it's very difficult to get individual level genotypes, which are traditionally used in population genetics. Because the marine environment has very diluted DNA in the water column, stochastic detection of rare variants is a problem. Read abundance, so how many copies of that gene I get after sequencing, is not easily equitable to the true frequency in the environment, and that's because it's not super well understood how different shedding rates, transport and degradation of DNA in the water column works. eDNA so far is commonly we use primers, which introduces a primer bias, as well as it has typically focused on the mitochondrial locus because it's abundant in the water column, or it's more abundant than the nuclear locuses. And then a little bit more I'll discuss on the bioinformatic trade-offs when you come to after sequencing. So my research question is, can routine eDNA workflows be refined to support inference beyond species presence in Solea solea? And I'll use two different workflows to explore this. One of them is the most commonly used in eDNA research, which is metabarcoding. I'll compare two different bioinformatic approaches, so data2 and denoise, and I'll ask if they differ in their suitability for CO1-based intraspecific inference. And metagenomic skimming, which is, I will compare more descriptively to library preparation methods, so the lab work. I will compare two brands, which is Illumina and Neb, and ask if their suitability is different in the recovery of Solea solea sequences, and discuss a little bit if these methods warrant further methodological development for applications in population genetics for marine fish. So to compare the two workflows a little bit, metabarcoding, the most popular, as I said, uses primers to amplify a target region or target locus in the genome, and metagenomic skimming, in contrast, sequences everything in the sample, but at a quite shallow depth, so it will detect mostly the most abundant sequences. Sensitivity-wise, metabarcoding is considered to be more sensitive because you are using PCR to amplify that target locus. And metagenomic skimming, this is still a bit of a limitation because my target species is a vertebrate and it tends to represent only a small proportion of the DNA that's in the water column. So you can waste sequencing powder on other things that I'm not interested in as much. For intraspecific resolution, metabarcoding tends to be limited by the short locuses that we target, and if you target a single locus, but haplotype detection is already possible in some extent. For metagenomic skimming, there's a high potential for the mitochondrial and nuclear genome because you will sequence everything, so you'll get multiple different markers, potentially a higher resolution, but nuclear recovery is still a bit difficult. So equating read frequency, so how many versions of the gene I get after sequencing to, was that exactly what was in the environment, is less reliable with metabarcoding. Because we use primers, they introduce a bias that's exponentially increased during the PCR process. Theoretically, metagenomics giving should be less biased because we don't have that primer bias and we just sequence everything as it appears. The biggest pro for metabarcoding is that there's large CO1 reference databases, which is the gene I'm looking at, and legacy data, while in metagenomic skimming that is still quite a limitation, is the reference databases. So for my metabarcoding methods, I will compare two denoisers, which are two algorithms used to clean error sequences out of our data and detect the true haplotype. So a haplotype is just the version of the gene that could be different between individuals of the same species. And an ASV is the inferred version of that at a single nucleotide resolution after denoising, which is the bioinformatic approaches. So artifactual sequences tend to occur in a lower abundance around a true sequence. And that occurs because of sequencing errors, chimeras, which is two sequences joining during PCR, and NUMPs, which is a nuclear mitochondrial pseudogene, which is a version of the mitochondrial gene that was integrated into the nuclear genome and is now accumulating mutations randomly, differently than the true version I was trying to target, which is conserved in the mitochondrial genome. So these two approaches, data 2 and denoise, have to have a trade-off where they do strict filtering, which risks false negatives or filtering out true low abundance sequences, or they can be permissive, which risks false positives or retaining artifactual sequences. So my workflow is the same for metabarcoding for both pipelines. The only thing I'm changing is data 2 and denoise, and they take different approaches fundamentally to how to clean out these error sequences. So denoise uses the Fred quality scores, which come from the sequencer, to create an error model, and then does a Poisson test to see if the abundance is as expected or not. And denoise is abundance distance deterministic model using entropy weighted, so it is a protein coding specific model. Everything else I did is the same, but here you can see my map of where the samples came from. I had 93 samples from 31 trawling events, so the filters, there was 3 per trawl, and in 2 known populations of Solea solea in the northeast Atlantic. For my metabarcoding methods, the bioinformatic workflow was exactly the same. The 2 things I changed was the lab work. So I used NEB and Illumina library preparation methods in the lab, and I only used a subset of 7 samples just to see if there was a difference moving forward for larger data sets. So the main results from metabarcoding are that both denoisers converged on the main signal after secondary filtering. So I started with over 1 million sequences, and after primary denoising, so the first set of error models, I was left still with a bit of divergence, so data 2 and denoise produced different number of Solea solea assigned sequences. Interestingly enough though, after secondary filtering, they converged a lot. So here you can see that they have 27 sequences, so they inferred 27 of the same ASVs. It looks like there's still a lot of divergence here because there's a decent amount of sequences that aren't in common, although when you look at the read counts, almost 99% of those sequences are part of the 27 shared. So the most abundant sequences, both pipelines agreed on. And they assigned reads very similarly within those ASVs. We were able to detect 8 of the 17 available references online, and I finished with 28 high confidence ASVs. That is the 27 shared, plus 1 of the references that was only in the data 2 pipeline. So some authors have proposed that using both models and the convergence is a conservative method for confirming ASVs. So when I did an analysis of molecular variants, I found no structure, but here you can see a haplotype network with 1 ASV in the middle, which was the most abundant, and very similar ASVs that only differ by 1 nucleotide around them, and then some more divergence ASVs. Most of them, you can see, are in both populations with 1 that was unique, but in general, all the ASVs were detected in all areas. So what's the reliability of eDNA-derived haplotypes? So, as I said, the denoisers agreed on the abundant ASVs, and secondary filtering is essential. This is consistent with the prior studies. Although the C1 fragment has limited resolution for population genetics, I still think it's a valuable resource for under-studied species, and it's an underutilized resource because we have a bunch of legacy data that we could be analyzing this way. So as I said previously, rare variant detection is difficult, but in this study I didn't have a large data set of CO1 from my study area, so I can't really tell you that I was able to detect or not detect the rare variants. The interpretation of the population structure, things to consider here are, I used incidence-based AMOVA, so I used presence-absence, and I discarded read frequency because of the issues I was stating before in the limitations. So it is known that incidence-based AMOVA can't distinguish true panmixia from a weak structure. So this should be taken into consideration that this population with other markers, higher resolution markers also, found weak structure between these areas. So potentially, other authors, so I did use the most conservative method in my paper, but other authors have suggested that read abundance can still have a lot of information and meaningful information. So there's a gradient of different methods proposed that should be considered, I think, if you have a weakly structured population like mine. And then a little bit now on the NEB versus Illumina library preparation methods. So that kits were functionally interchangeable, so the community composition was pretty much the same between them. The targeted sampling did improve read assignment, so typically eukaryotes and vertebrates are expected to be less than 1% in marine samples of the reads, and bacteria and other microorganisms would take up the bulk of it. So by sampling on trawls where we know the fish are, we were able to get higher read assignments. Hopefully that would translate to more genome coverage. So we were able to get up to 23% in some of the reads assigned to fish. So I think that's a useful method. And then for the mitochondrial genome, I think it was quite accessible. Most of our samples exceeded 90% coverage, and they had depths of sometimes up to 18 times. In contrast, the nuclear genome, as expected, didn't have as good of coverage, so I think it would be quite difficult to detect various versions of the gene when we only had 2 to 3 times depth the bases that we did have covered. So, in conclusion, can routine eDNA workflows support inference beyond species presence? I think partially for metabarcoding, depending on your research question, and for metagenomic skimming, not yet. So for metabarcoding, do data2 and denoise differ in their suitability? I expected that they might because, as I said previously, denoise is catered to protein-coding genes, which CO1 was. But data2 had the benefit of using Fred quality scores. So because of how different the methods were, I did expect them to be quite different. But they did converge on the main signal at the end. So I think that they both are quite suitable for this workflow. Is the signal quantitative enough for population level inference? I think partially. So we can confirm here that we can detect haplotypes and differences, different versions of the gene within a species. Although there's still limitations. Are we going to replace traditional tissue-based population genetics today? I don't think so, but I think if you had questions such as detecting an escaped aquaculture, ASD, or you were looking for specific haplotypes, I think that's possible, or it is possible already. And with a little bit more development, I think that if you had multiple or more variable markers to improve the resolution, there's still a lot of development that could be done to improve this method. I also would like to repeat that although there's limited resolution with CO1, we have a lot of species such as deep sea species or species that are endangered or difficult to catch. It's still better than nothing. So if you can't catch the individual and get a tissue sample, there's still a lot of value in using existing databases that have a plethora of underutilized intraspecific data from the CO1 gene. For metagenomic skimming, do the two library preparation kits differ? There was a small sample size, so that should be considered, and I use mostly descriptive statistics, but I don't think they do. I would base my decision mostly on what they cost and how easy it is in the lab for your lab if I was running a lab. And does the approach warrant further development? I think so. I don't think it's viable yet. We did have pretty good mitochondrial coverage, and I do think that there could be a discussion between Illumina versus long sequence nanopore sequencing to if we could define haplotypes easier one way or the other. I think nuclear coverage is insufficient, but the targeted sampling improved a lot from what we expected, and I think that there's a lot of enrichment methods that should be considered to improve the detection of the nuclear genome, which I think is very possible. And thank you. These are all the people that helped me through my project.
Thank you, Dara. We can start with the question. And what do you need the one?
Thank you very much for the presentation. I want to ask you how you decided which samples to use also for the metagenomic scheming.
So initially I actually used a subset of the original samples, but some of them failed. So we used some from 2025 and then some new ones from 2026 that weren't in the meta barcoding. So initially we wanted to use the same ones to see, the timeline for my thesis didn't allow to do population genetics, but if we had a little bit more time to be able to compare the samples directly, the meta barcoding and the metagenomic skimming, so initially we wanted to use a subset. I think this was mostly to explore where to invest, because with marine samples, metagenomic skimming isn't really used for this purpose. The lab kind of just wanted to test out the methods before investing in more. So the two sample, I don't think those samples would have been ideal if you wanted to do population genetics. They're very close. They're from populations we know that aren't super differentiated, but that wasn't really the research question. It was the research question focused on how many, does it differ and how many sequences we were able to detect from the lab work itself. So I think if you wanted to expand this, you would have to do enrichment for sure, but there's lots of methods that other areas use, like people that study bulk DNA from bugs or ancient DNA from museum artifacts. It's a bit different than in water samples. Again, you have much more diluted DNA, which is a huge limitation as well. But there are enrichment methods that work for other areas that could be developed for this. If you were going to do that, then I think you should choose probably a species we know has a stronger structure and samples that are a bit farther apart, and then of course more samples. But before investing in that, testing out some of the early lab work was necessary.
Yeah, I was wondering because maybe for comparison, like a gather sample that you know that have a specific structure, a specific kind, like maybe for aquaculture could help. I don't know if you consider something like that, like having I don't want to call it a black, that's something that you know that it's there.
I didn't explain in this, but we used a tissue sample as a positive control. So in my paper, I discussed that we can show that the method worked on the tissue sample because we did get great coverage and all that. So we did have a tissue sample that I didn't describe here. Also, something I explored a little bit in the report was the amount of fish in the trawl. So the samples did differ in how much Solea was caught. With only 5 samples, I wasn't able to do much statistics on that, but we did just visually see that it tended to be the trawls that had more fish also had more reeds assigned to Solea. So is that super conclusive? No, but it could mean it would be an interesting area to continue work on. Thank you.
Yeah, I will stay also with the methodology. It's not easy to. Yeah, but maybe a bit more question. I don't know why the CO1 gene was chosen, and because we could also go for some amplicon sequencing. I don't know the B3 B3 region that are more commonly used. What was the reason behind the choice of the CO1?
Yeah, the choice was, I agree. I also think that if you used longer or more variable markers, it would be more appropriate. But I think the goal here was to be able to integrate this workflow into something that was already existing. So it's cost effective if we have these, like the lab I work in is doing this sampling on a regular basis, and many universities and governments are sampling CO1. It's become an industry standard. Because of the massive reference databases as well is also quite helpful. So I think it was making the most of workflow that's already existing. I think that if you wanted to do kind of directed population genetics, choosing different markers, or at least more markers, would be useful, but something to consider when you choose longer markers, more variable markers. Primers sometimes aren't as reliable, and detecting them from the water sample is not as reliable, so the benefit of the short marker is everyone's using it, the databases are massive, and they're really reliably taken out of the water column. So on a monitoring basis, if it was something you wanted to integrate into existing infrastructure, which was the goal here, we used commercial trawls, so existing fishery, as well as BTS, so the fisheries monitoring trawls. So it was just a cost-effective way to integrate into that.
To change that, the marker you're using is depending on the organism you're looking for. I mean, now it's specific species, but in general, let's say someone wants to
Yeah, I think
use the same method.
if you're studying one specific species, you should use species-specific markers. Again, that's not super cost-effective if you're in a lab where people are studying many things, but I do know that the primers that we use aren't amazingly effective on elasmobranchs. So that's something that maybe I wouldn't use this data set to study. They are well proven with the fish, the commercially important fish in Belgium. So I think you're pretty safe to use these universal primers for the commercial fish that they've been studying already with that. But if you wanted to move into elasmobranchs, and then of course invertebrates and more divergent groups, using more species-specific markers would be more appropriate.
You mentioned about unique sequences, like there were quite some and so on. Like, were the singletons removed in your filtering protocol or not? Because you mentioned chimera filtering and so on, but maybe I missed or did not understand about the singletons.
So I didn't discuss so much filtering as much. Another thing I think would be very beneficial is when I was doing my literature review, it doesn't seem like there's a lot of harmonization in the workflows. People are doing things differently. And I think that there's still a lot of benchmarking to be done into where in the workflow you do chimera removal or what are the minimum abundance. So if I remember correctly, it is in my report, but I added a minimum abundance of, I think, 10 per sample, but I did sit there for a while and think about you could add a larger one for all samples pooled, or do you add a minimum abundance per sample or per area, and should you do the minimum abundance that's just a number consistent like I did, or as a proportion of the reads in that sample. So there's a lot of choices, and it seems like researchers were using different versions of secondary filtering, and I do think that it does affect your results a lot, the type of secondary filtering. Also within all of these, like data 2 and denoise are super comparable because they're different. They use different stringency parameters that can't really say are the same. So you can choose how strict and how permissive you want both of them to be with different parameters in the model, and you can try to choose something that you think is similar in the outcome, but they aren't directly comparable that way. And that also goes for Coil and Lulu. You have the freedom to define cutoffs, and I think harmonizing everyone in the eDNA workflows, that would benefit future research, because as I was looking, it seems like people are still at the point where they're exploring different options, and you do have the freedom even within the algorithms that you choose, but also between the algorithms you choose. So there's other options, like I decided to go with Lulu, but there's also MetaMate, which does a very similar thing, and different researchers are using different algorithms.
Thank you very much.
Thank you.
Yeah, sorry. Maybe just a short question. Maybe I missed it, but I'm not sure what are the samples physically. You talked about trawl nets.
Yeah, so the samples here, each, so I have 93 samples, but I had 31 trawls. So we used commercial trawls as well as BTS trawls, so the Belgian scientific trawls. And on each trawl, we had a little circle that we call a metaprobe, and it's just a 3D printed plastic circle that has a gauze filter in it. So that's the targeted aspect of it, is we put the filters on the trawl went through, did its thing, we pulled them up, and the idea is, okay, well, we know there's lots of fish there, they're targeting Solea and other benthic species, so hopefully there's a lot of DNA in the water column to try to overcome the problem with DNA being very diluted in the water column, and the transport shedding degradation issue is compounding to that. So it was a cost-effective way of just like basically 3D printed plastic circles and put some gauze in them and tied them to the trawl. Other people in the lab were also exploring, can we just take the samples of the water at the surface and is that good enough? So there's other research questions being asked with these samples. They are doing water samples, so just the traditional way, take a water from also like the fish are still there, so it doesn't matter if you take it from the trawl or from the surface. And then also they were exploring automated sampling. So if we just put a box that just samples automatically near the surface as well. So I don't have the answers, that wasn't part of my research question, but it is a cool topic to think of. How can we integrate sampling cheaply into existing infrastructure? And this was in collaboration with commercial fishing as well, which is cool.
Okay, so it's quite limited to abundant and targeted species.
It's very for targeted species. There's another student here that was exploring if we can estimate abundance of the fish as well. Just eDNA.
Thank you very much.
Thank you.
I have a question. Thank you for the presentation. So you work with eDNA and you work in a marine environment which is highly dynamic. Do you have any idea how long it takes to break down this eDNA in this environment and how far it can be transported?
I think that is not well understood.
Yeah.
So there are people that are doing experiments in labs, and it's variable between species and the environment and how many currents are there, also how much sun is there providing UV to break down the DNA. So I think that is a major limitation. It would be so cool if I could just take water and be like, oh, 5 reeds, that means 5 fish. Great. But it is much more complicated than that, and it's different between different life stages of the same fish. They shed DNA at different rates. People have suggested that the level of stress the fish is under, or the species completely, obviously if you're looking at one cell versus a fish. And then the transport is also a bit complicated. So DNA can be transported quite far, so if you're getting DNA somewhere, it doesn't mean that that individual wasn't a couple kilometers away, but it's not well enough understood for me to give like a distance.
And is it possible that the absence of population structure is because just your eDNA is mixed from all populations together?
I think it is possible. I think that the targeted nature of the sampling would minimize that, but I definitely think it's possible that I would have to, I'm not super familiar with the oceanography of the region and how the currents work, but I know that's a huge factor. Maybe familiarizing yourself with that could answer the question a bit more. These regions are quite far, so I would be surprised if I could pick up the DNA here, but it's not something I explored in my research question. But it would be interesting. I think that is an area that we need a lot more work before we start replacing population genetics or traditional surveys with eDNA.
Thank you.
I think we can move to the
Yeah.
next one. Thanks.
Thank you.
Oh, can I try to set up the speaker view, or is that all right? Like the presenter view? Or would you
The
rather?
problem is with you cannot do the presenter view
Right, because you'll need.
because this will be streamed.
Right.
So
Okay,
it's okay.
it's all right. I have notes here.
Okay.
We can adapt.
We can.
Hello? Hello?
Second.
So, ecosystem restoration is not simple. It's a multi-stakeholder, resource-limited endeavor, and decisions need to be transparent and data-driven for success and sustainability. I'm Etienne Cancho, and I'm here to present my master's thesis, Multi-Objective Prioritization of Mangrove Restoration Areas Highlights Trade-offs among Biodiversity, Ecosystem Services, and Restoration Costs for Transparent Decision-Making. So my thesis was done under the supervision of Dr. Anthony Richardson and Elvis Debala from the University of Queensland, and Dr. Hanamad Villarreal Rosas from the Queensland University of Technology. So my thesis fits into efforts to enhance effective ecosystem restoration. So rapid ecosystem degradation has reduced biodiversity and its associated benefits, motivating ecosystem restoration. So one such policy for this is the Global Biodiversity Framework, an international agreement to enhance biodiversity and ecosystem conservation and restoration with goals such as to increase participatory and integrated spatial planning and to restore 30% of degraded ecosystems. So I particularly focus on mangrove forests. So mangrove forests are tropical coastal tree species that provide habitats for terrestrial and marine species and provide abundant ecosystem services such as carbon abatement and fisheries enhancement. However, they've experienced massive degradation due to drivers such as overexploitation and land use change, prompting international and national restoration efforts. So one such country involved in this is Indonesia, the country with the world's highest mangrove cover and the highest mangrove loss. Indonesia has set ambitious restoration targets to restore 6,000 square kilometers of degraded mangroves, necessitating strategic planning that accounts for opportunities, risks, and challenges. One such strategy to achieve this is spatial prioritization to guide restoration. So this is founded on different principles, such as the spatial variability of restoration outcomes. So for example, some areas might vary in the potential restorable biodiversity or ecosystem services. And improperly choosing the appropriate restoration sites could lead to missed opportunities or synergies, leading to reduced outcomes. So previous mangrove studies have used spatial prioritization to select mangrove restoration sites, but these have been limited by factors such as using only one objective for restoration, like only focusing on biodiversity only, or only one ecosystem service, which can miss the bigger picture of restoration. So to address this, other studies have used multi-objective optimization, which explicitly integrates different outcomes into the restoration planning process. And one advantage of this is that we can evaluate trade-offs and synergies when different outcomes are prioritized. While it exists for different mangrove studies, it has seen limited use. For example, many studies have considered only a few restoration outcomes, even though there are several, or perhaps they captured only a small trade-off space, leading to an incomplete analysis of different restoration actions. And other factors, such as restoration costs, which are vital for feasibility restorations, have not been included. So if multi-objective optimization is advanced and enhanced, it has the potential to increase the transparency of decision-making for more participatory and effective restoration. So given the need to restore mangroves and the availability of spatially explicit data on mangrove restoration outcomes and novel tools for spatial prioritization, we want to advance multi-objective optimization to further analyze trade-offs and synergies in mangrove restoration outcomes, specifically generating alternative restoration plans that can be compared and analyzed. So specifically, we asked for the forests in Indonesia, how can biodiversity, ecosystem services, and restoration costs be integrated into a single multi-objective prioritization approach for mangrove restoration? And two, what trade-offs and synergies are evident among different spatial restoration plans in terms of site selection, biodiversity, ecosystem services, and costs? And corollary to this, we also asked what drives these trade-offs and synergies. So to start, we define our study area and we use spatially explicit distinct patches of mangrove forests known from Indonesia. These are available from previous literature, and these patches are also further subcategorized into different geomorphic settings of mangrove forests. So these are influenced by the biophysical factors in different mangrove settings, and each unit is categorized into deltaic, estuarine, a laguna, or an open coast unit. And for each unit of mangroves, the amount of restorable area is also known. Now, each planning unit has data on two ecosystem services, one being carbon abatement by restored mangroves in soil and biomass over 40 years, fisheries enhancement, which is the number of additional individuals of commercially important fish and invertebrates after restoration. And we also have information on biodiversity, which is represented as the amount of area in each restorable mangrove unit that intersects with the distribution of mangrove species. Now, in order to also capture not only taxonomic biodiversity, but also ecosystem biodiversity, we further segregate these mangrove species into their geomorphic settings. And lastly, we also have, again from previous literature, information on the estimated amount of restoration costs to completely restore the mangroves in a given mangrove unit, expressed in 2022 US dollars. So we use an R package, prioritizer, which explicitly was made for spatial optimization and systematic conservation planning. So this package, given a set of parameters, goals, objectives, and targets, can select spatial units that optimally meet restoration outcomes. So first, we created 3 single objective prioritization problems. One, to maximize biodiversity gains, and we set for this a target to restore 30% of the restorable area of each mangrove species in line with the Global Biodiversity Framework. One, to maximize ecosystem service gains for carbon abatement and fisheries, and we set a target of 100% for each service, effectively maximizing each. And another single objective target to minimize restoration costs. So it selects the planning units that are the cheapest to restore. So each single objective problem using a new functionality in Prioritizer was solved hierarchically in a multi-objective fashion. So the first problem solved was the biodiversity problem, followed by the ecosystem service problem and the restoration cost problem. And we also found solutions that are also limited by another parameter, which is to limit the restorable area to 30% of the total restorable area, in line again with the GBF targets. Now, we also analyzed trade-offs, specifically focusing on biodiversity and ecosystem services by creating alternative restoration plans. Now, these alternative restoration plans still factored in biodiversity and ecosystem services, but we varied the amount to which each outcome was targeted. So with that, we created 139 different spatial plans, and we selected 3 in particular for comparison. So one is the solution to maximize biodiversity, where selected units are in green and the unselected units are in gray. Similarly, we chose another one to maximize ecosystem service gains, and we also selected another one, the equal trade-offs solution, which balances restoration of biodiversity and ecosystem services. So when we look at the spatial selection of these different units, we see clear differences. This map shows which units are selected by which solutions. So some units are selected by only one solution or the other, indicating differences, while others are selected by several, either 2 or 3, indicating similarities between the different solutions. But we can also quantify the level of agreement between the solutions using a metric, Cohen's kappa. So this measures essentially the level of agreement between raters, and in this case, it's the solutions. So we see that for the biodiversity and ecosystem service solutions, they have a very low agreement, where 0 is agreement by random chance. And we also see slightly higher agreement with of the equal trade-off solution with the biodiversity and the ecosystem service solution. Now we also see variations in these solutions in their biodiversity, ecosystem service, and costs. So this is a parallel coordinate plot where each line is one of the 139 restoration solutions. Each axis is a restoration outcome, and the intersections indicate the values of each outcome for each solution. So let's look at the biodiversity solution. It has the highest biodiversity restoration, but it also has the lowest ecosystem service restoration, and also happens to be the most costly solution to restore. We see the opposite for the ecosystem service solution. It has the highest ecosystem service restoration and is the cheapest to restore, but it has the lowest biodiversity restoration. When we look at the equal trade-off solution, we see a balance. We see relatively high biodiversity restoration, moderate ecosystem service restoration, and relatively a lower cost for restoration. And while we point out these 3 specific solutions, we note that we generate a range of different solutions. And when presented to stakeholders involved in restoration, any one of these could be a potential solution based on the preferences and local knowledge of the stakeholders. We also notice a difference in the geomorphic setting of the units selected by each solution. So in particular, we pay attention to the amount of deltaic and open coast mangroves selected in each solution. We see that the ecosystem service solution on the right has the highest amount of deltaic mangroves while having very little open coast area. But as we increasingly prioritize biodiversity restoration, the deltaic area decreases in favor of more open coast restoration. So with all of this, we assert that a multi-objective optimization approach can enhance transparent decision-making and trade-off analysis for mangrove restoration. So there you go. So restoration involves different stakeholders and goals, and many of these can be conflicting. And if we fail to include these different goals and the trade-offs and the expansive trade-off space between them, we could ignore synergies or make inefficient decisions leading to suboptimal outcomes. Now, by explicitly integrating and quantifying these multiple objectives, we can create decision spaces that are more accessible and easily understood by different stakeholders, thus enhancing the transparency and agreeableness of different solutions. These solutions can be deliberated, debated, and discussed by different stakeholders, and that can make decision-making more inclusive, more transparent, and more effective. Now, we don't intend for our tool to identify single best restoration outcomes alone, but rather, we want to give stakeholders and decision makers the opportunities to really deliberate and discuss these outcomes, because restoration outcomes are very dependent on laws, local knowledge, and different circumstances. So in terms of restoration, what you really want to achieve is flexibility and openness. Now, we also emphasize geomorphic setting as a driver of trade-offs and synergies among these three studied outcomes. We particularly pay attention to the balance of deltaic and open coast mangroves. From previous literature, we know that deltaic mangroves have higher productivity due to riverine inputs, and higher productivity leads to better growth, more carbon abatement, and enhanced fisheries. Riverine inputs also increase sediment into coastal areas, which accelerates mangrove restoration, and this makes mangrove restoration more cheap. So we see a synergy of deltaic mangroves being higher in ecosystem service restoration and lower in cost. Now, the opposite is true for open coast mangroves that don't have as much, if any at all, riverine input. And we see that the open coast mangroves are more represented in the biodiversity oriented solution because of the way that we set the restoration targets. Again, not only are we enhancing taxonomic biodiversity, but also ecosystem biodiversity. So if you want to restore more diverse ecosystems, you have to include these less efficient different geomorphic settings. So that leads to an increase in restoration costs and a decrease in ecosystem services. So we point out some caveats that constrain the findings of our results, of our study. One is that we use global scale data for analysis because currently this is the most complete and available for mangrove distributions and ecosystem services. But local scale data could be more accurate. Second, we set uniform species restoration targets of 30% for every species, but this could change if rarer or more range-restricted species are given higher targets. Third, we also assume full restoration of mangrove units, but in practice, this might not be feasible, and only portions of units might be restored, thus leading to different restoration outcomes. And we also consider that we might underestimate the restorable area because our data is based on satellite data from 1996 onwards, even though extensive degradation occurred prior to this. Regardless, this is still the most available data set we have on mangrove extents. For future directions of our work, we recommend integrating other mangrove features, such as other ecosystem services or sociocultural valuations if these data are available. And we also recommend the application of this approach to different scales, especially at the subnational scale, where more productive and direct communication between stakeholders can take place. So in summary, we find substantial trade-offs and synergies between biodiversity, ecosystem service restoration, and restoration costs for mangroves in Indonesia. And we also identify geomorphic setting as a driver of these trade-offs and synergies. We also note that balancing trade-offs can lead to optimal solutions that account for both or for multiple restoration outcomes. And overall, we assert that multi-objective prioritization can increase transparency of decision-making for more effective, sustainable, and impactful restoration. Thank you so much for your attention and your time, and I'm happy to answer any questions you might have.
Okay, thank you. We can start with the question. Anna, would you like to start?
Yes, I can thank you for your presentation. You brought us to another environment.
Different field.
Very nice. Yeah, you mentioned a couple of times in your presentation and in your research course, the different types of diversity we're targeting for the restoration process. So if you're looking to taxonomic diversity or ecosystem diversity, for example, how do you define that exactly? How do you define ecosystem diversity?
So actually, I have a hidden slide. Can I show that? Okay.
Yes.
So there. Okay. So the decision to capture all ecosystem service diversity in addition to taxonomic biodiversity is based on previous studies of mangroves that are not only in spatial planning but also attempts to characterize the mangrove ecosystem in general. Mangroves existing along coasts are subject to different biophysical factors that are variable across coastal areas. So many authors in the past have attempted to characterize these ecosystem differences, and that has led to this typology. This is the most recent typology produced by Worthington a few years ago, and it basically summarizes the extent of the main drivers that segregate and categorize mangrove ecosystems. So deltaic, estuarine, lagoonal, open coast. And as you can see, they're subject to different coastal forces, and we define these as such because global biodiversity initiatives have explicitly integrated these multiple definitions of biodiversity into restoration processes. So biodiversity as a whole is composed of genetic biodiversity, taxonomic biodiversity, and ecosystem biodiversity. So our approach attempts to capture two or three of those. So essentially, for example, let's say mangrove species X. Mangrove species X can occur across Indonesia. But let's say in a given mangrove patch, that is defined as a deltaic mangrove. So all of the mangrove species X there is deltaic mangrove species X. And then units of, let's say, lagoonal mangroves, that will create a new subspecies called lagoonal mangrove species X. So this is a method that has been done in previous mangrove restoration conservation and restoration studies. And given the available data, that is one way of defining biodiversity. We do recognize that there are other factors that play into defining biodiversity, of course, like the genetic component, as I mentioned. That data is not available yet, but in the future, that would be useful in creating more encompassing and accurate definitions and plans for restoration. Does that answer the question?
Mm-hmm.
Okay.
Yes, and then, for example, because now you are explaining us about the characterization based on geomorphology, for example, maybe habitat suitability for more species, things like that.
Yes.
Are there multiple ways to define the ecosystem functions and ecosystem services, of course? Okay. You mentioned also restoration targets and where you put the threshold there. It also depends on the situation, country, different stakeholders, and so on. What would you think, from your experience in your study, what would be, let's say, your own recommendation if you were called to consult, let's say, the local community there, or the politicians, or the stakeholders that are trying to define their own strategy? What would be your own recommendation?
I think one thing that would definitely improve the recommendations of the study is to change those targets for biodiversity to prioritize rare species. So, for example, if you have a species that is critically endangered by IUCN standards, for example, you could set a restoration target that's much higher, like let's say restoring 90% of the degraded area, and more common species, especially those that exist in other countries as well, or those that aren't endemic, could have lower restoration targets. The only problem with that is that it is also a very subjective process. There have been ways that have been recommended to standardize the target setting process, such as using logarithmic methods for defining higher restoration targets for rare species, but these methods are, again, setting these targets is very context specific. So that's one way I would regardless of the difficulties, I think that's where I would start. Try making the targets more sensitive and aware of different taxonomic discrepancies.
Thank you very much.
Thank you, very nice presentation. I was wondering why, when you saw different strategies, why every time that you select the ecosystem service, your diversity go down? There is no connection between the ecosystem service for a specific diversity of mangroves?
Can I just go back to that slide here? Is it this one? Yes. So the reason why biodiversity has a trade-off with ecosystem services in this particular context is because of the decision to include ecosystem diversity as well in the prioritization process. So ultimately, the most effective geomorphic setting that restores ecosystem services is the deltaic mangroves. Again, because of the geophysical factors that make mangrove growth more favorable, leading to more ecosystem services. But in this specific case, we also want to restore a diverse set of geomorphic settings. So that necessitates clearly, if you want more open coast mangroves for sure, then of course you're going to have to reduce the amount of deltaic mangroves that you have. So you're still restoring the same area of mangroves. It's just that because you're choosing to restore a geomorphic setting that's less effective at delivering ecosystem services, you have a trade-off in reduced ecosystem services. Does that answer the question? Or?
Yeah, I was wondering if you try to apply the model in case that, because I understand that this is because one kind of mangrove it's giving more services, but what happens if each kind of mangrove is giving a different kind of services? And then you can restore each of them for a different service.
Yeah. That definitely would make sense, especially at the subnational scale where the amount of ecosystem service per mangrove tax is known. But that is actually a gap that has to be remedied in the literature. There are differences between taxa, but most of the data available on mangrove ecosystem services is just applied at the plot scale, assuming all mangroves are the same. But I will get ahead of myself and say that there are differences, definitely, because mangroves vary in their structure, like their physical plant structure, and that changes habitat availability, storm attenuation, or the rates of growth can also affect how much carbon is sequestered. So unfortunately, that is a gap that exists because of the lack of data on taxon-specific mangrove ecosystem services. And that's definitely more important when we look at, for example, traditional uses of mangroves, like some mangroves are used for a specific type of, let's say, bark, or maybe they host other taxa, like honeybees that are harvested for honey. So that is something that doesn't exist in the literature as well. So in terms of data availability for mangrove ecosystem services, it's really very broad and at the landscape level. But if anyone is interested, taxon-specific studies would help make the process even more transparent and accurate.
Thank you very much for your presentation and your response. That was really interesting and insightful. As you just said, I think that this kind of approach is really interesting, but it relies strongly on the data that are available. And I was not sure regarding biodiversity, if you only integrate the biodiversity of mangrove trees or whole taxonomic biodiversity of the whole community.
So you also refer to like invertebrates, fish.
Okay, so it includes.
We're focusing solely on the trees,
Okay.
and that is another factor. I mean, the other trees might have stronger associations with biodiversity, but again, that data doesn't quite exist.
That's concretely how works restoration? How do you restore a mangrove?
So there are several methods to restore mangroves. The most popular way that has become very common is direct planting of mangrove seedlings.
Yeah.
So you find a muddy bed and then you plant seedlings there. That has been criticized because many of these areas have not been really suitable for mangrove restoration, and a lot of the species also selected don't last in those areas. Another method that's been gaining more traction is simply ecosystem, or rather hydrodynamic restoration. So what that means is restoring the um, hydrodynamic flows, like riverine inputs, or removing barriers in the areas, like whatever concrete structures, or for example structures for fishing that were previously there, and then allowing natural restoration to take place. And there are studies that show that natural, unaided in a way, restoration is effective. That if you just simply leave a place alone, the propagules of or saplings of mangroves will arrive.
Passive versus
It's passive restoration, essentially. So each one has its drawbacks, of course, advantages and disadvantages. So with conscious restoration, of course, you can achieve more quickly, but then there are biases, like you are in a way deciding what species colonize, and that could ecologically, in a way, you're disrupting the process of colonization, of succession. And yeah, well, anyway, methodologically, regardless of the outcomes, these data for ecosystem services are essentially averages or expected outcomes. Even these values have ranges. So these are recommendations, but they're certainly not concrete predictions that definitely if you do this, like, we're not saying that if you do this exact spatial configuration, you're going to get this much carbon, but it is a way to make that decision-making process more transparent and more understandable.
In your opinion, what makes restoration attempts successful?
Restoration is actually, in my opinion, it's more social than biological. Because when we talk about restoration, we have parcels of land that have been degraded, and they've been degraded for a reason. Normally, it's because of some kind of productive use, like farmland, or non-productive use, like buildings or any other infrastructure. And when you get degraded land and you want to restore it, not only are you inputting money to actually perform the restoration, but you could potentially interfere with people's livelihoods, with their fishing activities, or perhaps like even like something like a resort nearby that has been designated for construction. So, in my opinion, having this data very transparent, like having the numbers, can show that if you do this specific spatial plan, we can have this much quantified benefit. And is that enough to offset, or is that equal to whatever economic activity that would be there actually? Because restoration involves monitoring, assessment, evaluation, and repeated funding. So given time, any ecosystem that's left alone will return to some sort of natural state or will undergo succession. It's in fact, in my opinion, the anthropogenic influences that are to basically hinder complete restoration. So, in my opinion, the best way to make restoration successful in the long term is to make it compatible with human systems, and that really does involve multi-stakeholder conversations. So it's complicated, but not biologically, socially.
Thank you. Couldn't agree more. Very much.
Okay, thank you. Let's stop the session.
Okay, thank you.
Oh.
Is it not just?
No, because it's I have to go back there.
First
And then go, yeah, every student you have to do that. It's not a big problem.
I'm so happy you came to mind.
Yeah, I am my work.
Thank you.
I think that
jury is ready.
Okay,
Yes, I will give you.
yes. Welcome everyone to this seventh session of the day of the IMBRC annual symposium. So we'll start this session by presenting the jury member to you. So there is Anna Maria, that is a postdoc at the University of Ghent. We have Patricia Pinto, that is here. She's a CCMR at the University of Aargau. And Nicole Vrijberg is connecting physical oceanography and connectivity and things like that. And there is Helen Gonzalez, that is a researcher here at the Marin Station, Valencia. And there is myself, I'm assistant professor at Sorbonne University. So we will start our series of 3 talks by Elena. That will present us the evaluation of ocean warming and ocean acidification on Stramolita hemastoma population on the Canary Island. So Elena and 4 others presenting. I will move when you are close to the time, like by 1 minute or something like that. And then after your presentation, I will ask the jury member some question for 15 minutes.
Okay.
Okay? So we will be happy to listen to you.
Thank you very much. I'd like to welcome you to my presentation of my master thesis, where I was evaluating the effects of ocean warming and ocean acidification on Stramolita hemastoma populations across the Canary Islands. And I'd like to begin by showing you these maps. And they show how ocean acidification is going to impact our future ocean by the end of the century. So the first map shows that the pH is only declining a little bit. The second map represents the intermediate warming scenario with high degrees of warming, and in the lower cases you see the high emission scenario and the very high emission scenario, where the impacts of ocean acidification become more pronounced by the end of the century. But to understand this process, we need a little bit of a background in chemistry. So imagine that the ocean acts as a carbon sink, and CO2 emissions, they dissolve into the seawater, and then they form carbonic acid. And this carbonic acid releases hydrogen ions that make the water more acidic. Usually, the ocean is in an equilibrium, but as the CO2 emissions increase, there's also more hydrogen ions available that make the water more acidic. And this might become a problem, especially for calcifying organisms, because carbonate ions bind to those additional hydrogen ions, and then this carbonate ions is less available in the water, which means that especially calcifying organisms are very sensitive to these changes. And next, we move to a specific site, my study area, the Canary Islands. They are located approximately 100 kilometers in the Northeast Atlantic Ocean, and they are considered as a marine biodiversity hotspot as they host a lot of endemic species. And as you can see here, we have a gradient from cooler waters in the east towards warmer waters in the west. And also, the main islands consist of Lanzarote, Fuerteventura, Gran Canaria, Tenerife, La Gomera, La Palma, and El Hierro, as well as the Junijo archipelago consisting of several smaller islands. But the climate has been changing in the Canary Islands, so the sea surface temperature has increased in the past decades at 0.2 degrees per decade, and additionally there was a pH decline of 0.05 units since 1995, and marine heat waves have become more frequent and more intense, which has been associated with biodiversity loss. For example, there was a big mass mortality event of a sea urchin species, as well as the loss of macroalgal forests. now we want to quantify environmental stress. And a good method to quantify this is measuring the metabolic rate, which is the rate at which an animal takes up and transforms and expends energy. It is also determined as the pace of life, as it determines growth, survival, and reproduction. I selected Stramonita henosoma as my study species because it is an intertidal predator that is very abundant in the Canary Islands, across all islands. It inhabits the mid and low intertidal zone, and it is a predatory species. It is protected since 2011 and before it was harvested and consumed by humans. So the main aim of my study was to assess how ocean warming and ocean acidification influences the ecology and the physiology of the species. So first, I wanted to see whether environmental variables, for example heat waves or sea surface temperature, influence the abundance and the body size of the species. And second, I conducted lab experiments to see what are the physiological responses. So I measured the metabolic rate. Now let's head to the methods. This was the field study that was conducted between 2022 and 2025, where they collected a total of 2,770 individuals across 91 locations in the Canary Islands. The black dots represent all of the sampling locations. Then first abundance was assessed using 3 replicate transect lines parallel to the shoreline, and every individual was counted as well as the shell size. It was measured, resulting in a data set of size and abundance data. Then I wanted to characterize environmental data to see whether it influences the size and abundance of the species. So what I did was downloading Copernicus marine data from sea surface temperature, first from a historical decade and then from a recent decade, to identify anomalies, which means areas that have warmed faster compared to other areas. So yeah, for the sea surface temperature I used this anomaly, and I did the same for pH. Then I conducted generalized additive models, including the variables, spatial locations, heat waves, the maximum sea surface temperature anomaly, and the pH anomaly. So both rasters that I did, where I extracted the exact value from all of the sampling locations. And since only sea surface temperature was the only relevant variable, these were my final models. Well, now we are heading to a very specific site in the north of Tenerife. It is called Punta de Hidalgo, and it represents a typical habitat for the species. And I collected 108 individuals during low tide from the low and mid intertidal zone, and then I transported them to the lab together with water from the collection site. Once in the lab, the individuals were acclimated in acclimatation tanks looking like this. So I separated them into individual containers together with some substrate to reduce stress. I controlled both the pH and the temperature, and individuals were not fed during this time because we didn't want to elevate the metabolic rate due to digestion. I had 4 treatment groups. So the first group I would treat with high temperature, 26 degrees. The second group I would treat with a low pH, and then I combined both stressors, and I also had a control group. Next, after the acclimatation period, I put the animal into a chamber. The chamber is supposed to be here, and now it looks like a little bit different, but I will explain it. So the chamber is submerged in a bath, which is here. Imagine there's an aquarium, and it has the same conditions as the acclimatation tank, and then the system is able to alternate between 2 modes. So first we have the flush mode, where the water is taken from the water bath, and then in an open mode. So the oxygen levels are restored to 100%. And then the measurement mode follows, where the decline in oxygen caused by the animal is measured. And this is repeated 3 times, resulting in a total of 41 minutes of measurement. In the meantime, we were able to control all of the parameters in real time, including the dissolved oxygen that is measured by the dissolved oxygen probe, and this is the pump that pumps the water into the chamber. So this method is completely non-lethal, and after the measurement period, the animal could be placed back and brought back to the wild. This is just to show you some variables that are important to calculate the respiration rate. We also accounted for the respirometer volume as well as the volume of the animal and the animal weight. And before every 2 to 3 individuals, we measured background respiration because we were assuming that microorganisms are also consuming oxygen, and we wanted to correct the final oxygen consumption rate. Now we are looking at the results from the GAN model. So the first map I got is this one, and we can see that areas in the east are warming faster than areas in the west, indicated by red areas. And especially around Tenerife, we even observed negative sea surface temperature anomalies, which also shows that warming is not equal. And the abundance decreased with increasing sea surface temperature anomalies. And next we're looking at the size GAN, and we can see that it showed a nonlinear pattern. So the shell sizes were the biggest at intermediate anomalies. When the water was warming around 1 degree Celsius, the shell sizes reached their peak before declining again. And now we can have a look at both maps together, because the first one shows or represents the mean sea surface temperature, and the second one is sea surface temperature anomalies. And we can see that areas that are historically cooler are warming much faster compared to areas in the west. So these might represent warming hotspots. And also, since only sea surface temperature anomalies was significant parameter rather than marine heat waves, we can also say that the rate of warming might be more, that the species might be more sensitive to the rate of warming than to absolute temperature itself. But the Canary Islands are highly heterogeneous and there's different habitats, so this might also contribute to the variability, and the models did only explain around 30 or 40% of variability. Next, we are looking at the results from the lab experiments. So as you can see here, there's the oxygen consumption rate, or the VO2, and then we have all of our 4 treatment groups. So the first group was the control group, and the oxygen consumption rates were relatively low in comparison. And then when we acidified the water, the oxygen consumption was significantly higher, which makes sense because it is a stressor. Then warming increased the oxygen consumption the most, and interestingly, when we combined both stressors, the respiration rate was lower than in the only warming treatment, which means that there was kind of a suppression. Another very important covariate was body mass, because across all treatments, the smaller individuals exhibited a higher mass-specific metabolic rate than larger individuals, so they were consuming more oxygen. Next, I calculated the thermosensitivity, which basically describes the rate at which the metabolic rate is changing when the temperature increases by 10 degrees Celsius. And at an ambient pH, there was a very strong increase in metabolic rate with warming, but as I already mentioned, when the pH was lower, there was kind of a suppression, and the metabolic rate didn't increase as much anymore, which shows that the acidification again reduced the thermal sensitivity. And we found evidence on natural CO2 vents on La Palma, because after conducting the lab experiments, we went to a site where there is a natural CO2 vent. So due to volcanic activity, the water is naturally acidified, and the population that lives there is like living in our future oceans. And so we replicated the same experiments with the specimens from there, and we found a significant differences between the vent populations and the previous data that we collected on Tenerife as well. We had also a control on La Palma. But there was a significant difference between populations, and the vent populations did not seem to be that temperature sensitive as the control population, which was highly interesting. So for future research, it will be very interesting to understand whether these population differences result from phenotypic plasticity or from local adaptation. And what we did was studying the population at natural CO2 vents on La Palma, but it would also be very interesting to compare west and east populations, because we know that there is different histories regarding the sea surface temperature. And of course, in the end, we need genetic studies to know whether these differences come from genetic differentiation. Now, to summarize everything a little bit, we know that body size influences the metabolic rate. We also know that temperature is a very strong driver, but we did not assess sex differences because this can also influence the metabolic rate, but we did not want to sacrifice any individuals, so we could not include this parameter. We also did not control salinity because we collected the same sample, we used the same water from the collection site in our lab, and we did not. We were only looking at pH and temperature. Individual stress can also contribute to variation, and the pH was relevant in the lab, however it was not in the field. And time can also contribute to natural variability, but it was not significant in our study. So finally, to conclude, temperature was the main driver of population patterns. Sea surface temperature was linked to abundance and size. And in the lab, we found the temperature and pH influence the metabolic rate, but when the pH was lowered, it suppressed the metabolism. And even though Stramonita is very stress tolerant, it does not mean it is resilient to climate change. And also remember that the future is not determined yet, and it still is in our own hands. Thank you very much for your attention.
Thanks, Elena. Thanks for rushing a bit so that you won't go too much over the time. So I will ask the jury member to question you a bit. So, is there anyone that wants to start, or are we starting with you? Yeah? Okay.
Microphone.
Oh, yeah. And maybe we can do like 3 minutes for each sessions or something like that.
Hello, Elena. Thank you for the very nice presentation. Very well explained experimental setup. Very interesting study. Yeah, I would like to ask something. We'll start with the methodology, for example. You acclimated the animals for 24 hours before starting the experiments.
Yes.
I have 2 questions related to that. Do you think 1 day is enough for this acclimatization from the field to experimental setup, just to compensate for the experimental enclosure effect? And second is whether you consider the non-feeding for 1 day for these animals, if it is stressful, like it adds another stress for. I understand, of course, why it's needed for the experiment, but.
Regarding your second question, in nature, the species only feeds 2 to 3 times per week. So 1 day without feeding is not very much. So I don't think this has contributed to any more stress. And regarding the acclimatization period, due to time constraints, we could not
do any longer acclimatation periods. But I have read that the species might even be able to acclimate to warmer temperatures when this period is prolonged to 7 days. But yeah, we followed other studies and used similar acclimatation periods like that. So
But ideally, let's say you have the time you want for such an experiment, how long would you first run the experiment, and if you would adjust the acclimatization period?
I would do trials to test whether there's differences. So first acclimating them 24 hours and measure the metabolic rate under normal conditions, and then second do the same but with an acclimatation period of 48 hours, for example, or 72 hours, and to see whether there's differences.
It's very, very important. Yeah, I was wondering, you focus your physiology parameter that you study is metabolic rate. If you would have, let's say, this project for longer time or bigger scale experiment, what else would you focus on to again measure physiology, but in a more let's say in a better way, more parameters, more like I mean in other words, like is it only metabolic activity enough to test physiology response?
There's many different factors that we can look at, for example, calcification rate, or also juveniles are more sensitive to ocean acidification. So if we look at that, we might get also a better insight. And also measuring metabolic end products could be a way to assess how the energy is allocated, because we did not know that yet. We know that that a low pH has reduced the thermosensitivity, and maybe the energy is allocated to other to somewhere else. And yeah, we could also study that. It's true.
Thank you very much.
Hello, thank you so much. It was very good presentation and everything very well explained. So which brings me the questions, and my main question was already said. That was other kilometers, but I'll move to the, because I'm always curious on comparative physiology, always curious of what explains the overall behavior, or if you will do long-term experiments, for how long could you run? And which parameters you could use on the exposed animals, and then also if you would go for the ones of the nature, parameters related to maybe on reproduction, or you tell me if it will be a long-term exposure to
if it will be a long-term
notion.
exposure. I think it would be interesting to also do repeated measurements, but we were trying this before and it seemed to cause a lot of stress to the animals. So what we did before doing, now we did a linear model and comparing groups and only measuring individuals once, but before we had another kind of design where we exposed them and then we measured the metabolic rate, and the other day we would increase the temperature and measure again, and then we would decrease it again, but it was, the stress level was still very high. But maybe there's a way to do it without increasing the stress levels.
And how did you note the stress levels or measure the stress levels?
Yeah, we only measured the metabolic rate, but it remained high even though the temperature was lower.
And then extending a bit more to reproductive parameters or behavior, what do you think that you know about these species that could be measured, or circulating levels?
I think it will be interesting to do reproductive studies to collect egg clusters from the species. And also I think there's a lot of potential at CO2 vents that we have on La Palma. So studying, or especially focusing on those populations, can tell us a lot about the species or how the species might respond to climate change in the future, and or especially to ocean acidification. And yeah, we could collect egg clusters from volcanic CO2 vents, for example, and bring them to the lab and show and see how they develop, or if they have the same reproduction rates that individuals from normal water.
Okay, so that was clear. I don't have many questions. No?
Okay, thank you very much. Well, congratulations for the very nice presentation. I really enjoyed it. Everything was very clear, and we understood the whole experimental design and the results. Following the questions that were presented before, do you think is there any alternative to the repeated measurement design? If you want to do something like that.
An alternative?
Yeah.
Like the alternative, we did the alternative. It was measuring each individual once.
But if you want to track them in time.
I'm not sure if I get the question.
You could, repeated measurement analysis is statistically complicated.
Yeah.
But you could make treatments, and each of them you measure the animals once, but at different times.
Okay.
So you add a new factor that is timed with several levels from 0 to I don't know how many days, or that could work. And is there any other variable that can indicate some shifts in biological fitness that you will measure in this animal? It could be not very difficult to do. And I'm just thinking, these guys are carnivores, right?
Yes. Oh yeah. Studies on feeding and feeding rates.
Yeah.
Yes, of course.
Yeah, let's see if they feed so well when they are stressed or not. To see if those, because physiology is very important, but if you want to go beyond that, you want to see if the changes in physiology are translated to changes in fitness. Reproduction and also feeding could be important. And regarding the physics of your study, can you go back to this map of the trends of temperature and pH in Canary Islands? Those were very interesting. This. So, how did you get the anomalies? It was just subtracting, no? The two different periods.
Yes, I was downloading average data from historical decades, so from 10 years in the past, and then from 10 years in the recent past. And then I extracted both. So now we can see areas that are comparatively warmer and areas that are comparatively cooler.
Yeah, it's very interesting. You could have done a proper gamma analysis with each trend at each pixel, separating the seasonality and the linear trend of increase, and give a trend in degrees per decade or something to see if it compares with that. Because I've done similar analysis in that area a little bit closer to the coast of Morocco, and I see cooling there. I see cooling to the west.
Yeah.
Because the upwelling there is getting stronger. Did you do this analysis also for other areas around, or you just focusing that?
I was just focusing on the Canary Islands.
That's very interesting. And just my last question would be, because you have all these points, these sampling points in space, you take into account pH, and you take into account surface temperature as your predictors. But this is a spatial analysis, so space is also important.
Yeah.
Would you add another factor to account for that?
Yeah, I think I included spatial location.
Okay.
Yeah, spatial location was included, but it was not significant.
But how did you account for spatial location as a factor? or as coordinates?
As coordinates, yes.
Ah, okay, that's good. I didn't get that from the
Yeah.
That's good because that tells you the spatial autocorrelation.
Because, for example, with another species, there was a correlation.
Yeah.
With of special location.
You could have one place
Mhm.
that you cannot explain all the variation because of the SST or pH,
Yeah.
but just because besides that place there is another one with the animals behaving similar.
Yeah.
That's because they are close. Okay, thank you very much for your answers.
Okay, Elena, thank you very much for your presentation and for all the discussion so far. It's going great. So this is the last one. So I was wondering during your defense, especially on the laboratory experiment with something that Nico just mentioned. So it didn't remain clear to me for how long were you measuring these guys' breathing?
First, we acclimated them for 24 hours.
Yeah.
And then I conducted the actual trial where I was measuring the metabolic rate, and this lasted for only 40 minutes. And after the animal was done, and we could release them back into the wild. Also, my survival rate was 100%, so no individual died.
So you only measure once.
I only measured once.
Do you think that there will be differences, for instance, during the day? So are these animals like night crawlers, or are they daylight people, or?
I read that they're mostly active in midday and the afternoon, and I also tested for that. So when I started the trial, I was also noting the time of the day, and I was testing for significance, but I could not find any significant trend in my case.
Mm-hmm.
And maybe also to mention, I was measuring between 9 a.m. and 5 p.m. every day.
Okay. Yeah, because that could maybe in the most sensitive times there is some higher trend or fastest increase. Let's see. Also, regarding you show a graph with the relation between size and temperature. Could you go to that? Yeah, before.
Sorry.
Okay, so this is for size, and the previous ones is for abundance, right?
Yes.
So, can we, because it would have been nice for me to look at them at the same trend, because do you think that abundancy is also maybe also connected to size? So if there are bigger animals, there is more competition, so probably there are less animals, or do you think that those two can be connected and can be also are treated as confounding factors or are correlated factors, or?
Yeah, I think sizes and abundance are correlated. So at intermediate abundances, there are also intermediate sizes, I'd say.
Yeah, and why would you expect, like, at intermediate temperatures, there is the highest growth or bigger animals, biggest animals? Why do you think that?
They seem to have an advantage. Bigger individuals, they seem to be more resilient against intermediate warming. Or this could also represent like the optimum range of the species when warming.
Yeah, it could be an ecological factor, something like food availability or something like that. Yeah, it would have been nice also to have, for instance, that, right? To, like, I don't know what they feed on, but also looking at this ecological, yes, some extra thing just to figure out. But it's not my expertise, so it's just my crazy idea. So I think I'm done. Yeah.
Okay.
Thank you. Thanks a lot.
Thanks a lot. We now move to Lola is next. Yes, Lola. Just before lunch, so that we, you,
I hope you're still alive.
yeah, you are our appetizer.
Yeah. Can I use the timer?
Of course.
Yeah.
Yeah.
Cool.
It's like this.
Okay.
These are your ears. Good.
The problem of being a star. Okay.
Your
Okay.
press this one to go forward, and you can still go. Okay.
Okay, so Lola will be presenting her work on the regional variability in the trophic ecology of Iliad Quaideti.
Good morning, everyone. So I'm Lola Jean, and I'm happy here to finally present my master thesis on the regional variability in the trophic ecology of Ilex conditi. So we focused on Ilex conditi as is a key ecological player in marine food webs. So as mesopredators is connecting lower trophic level to top predators, so it's really a trophic bridge. It's also a fisheries target in European water and mostly in the Mediterranean Sea. Is widely distributed, and among the European waters is really widely distributed. So because it lives in such different environments, we wanted to know if its trophic ecology were different across all of these regions and across also different life stages that I will explain later how we did that. So we sampled in the Spanish Iberian waters with acoustic survey in 2024 with a standard protocol. So the first region was the South Bay of Biscay in spring, the Gulf of Cadiz in autumn, and the Western Mediterranean Sea in summer. So we used the pelagic trawl, and when we had the individuals, we took two different tissue from the individual, and the muscle tissue from the dorsal mantle part of the individual and his eyelenses. And using these two tissue, we processed to stable isotope analysis. So I don't know how much you know about stable isotope analysis, but it's summed up by a simple principle that summed up by De Niro and Epstein in 1976. So you are what you eat plus a few per mile. So basically, with isotope, we can know the trophic position of the individual and its foraging habitat using isotope ratio of nitrogen for the trophic position and carbon for the foraging habitat. So I used two different muscle tissue. So the first one is the muscle, as it's a metabolically active tissue with a active turnover about weeks to months. Analyzing this isotope within this tissue show the diet of the individual around over a recent period compared to the eyelenses that show the diet across its whole full life cycle. So representing a chronological trace from birth to capture as its inert metabolically tissue. As eyelenses are a bit new in trophic ecology, I wanted to explain a bit more on that, so I focused mainly on eyelenses on this presentation, and I did a little animation to explain where these eyelenses working. So, okay, you need to kind of imagine the eye like that. So like a ball, and so if you cut horizontally, what you will see here will be the plan view on that. So when the squid just born, the baby squid is just, he will form his first layer, and as I said, it's inert tissue, so it will keep the trace of the isotopic signatures in this layer. So the core layer represents the born event of his diet at this time of his life. Then as it grows, you have another layer, so another isotopic traces in this layer, etc., etc. So another layer, another time period, and another isotopic trace that is keeping this layer. until the time of capture or the death event, which would be the outermost layer. Obviously, this is really schematic. You have to imagine it with a lot more of layer and with a lot of more methodological limitation. So we only choose to analyze the core layer, an intermediate layer, and then the outermost layer, because we could not analyze all single layer of all single individual. And from that, we expected the nitrogen, so the trophic position, to increase with size or age as bigger individual, bigger prey. The same for carbon, because it can go deeper. And because we expect these two, we expected the isotopic trophic niche to be bigger across its life stages. And I think with a bit of muscle here, comparing the muscle tissue isotopic signature with the islances, the outermost layer of the islances, we expected a relationship 1.1, so that we could compare this to to have an overview of his diet. So the first result is the isotopic trophic niche. So you have in each square. So in each square you have the Bay of Biscay, the Gulf Coast, and the minerality, so each region, with the isotopic space, so nitrogen and carbon. And here the range is expressed in micrometer. And if you follow the little animation before, here you have the core layer, so the born event, the intermediate layer, and then the outermost layer. That changed for the Gulf of Cadiz because we estimated the ranges from complete isotopic tracks of individuals, and they are different for each region. The Gulf of Cadiz had bigger individuals. So the first result was unexpected. As you can see, the red ellipses in the Bay of Biscay and the Mediterranean Sea is bigger than the outermost layer, so that we actually have a contraction of the niche across these life stages. So far, we don't really have a big hypothesis on it because it wasn't expected. The only thing is that trophic ecology is normally based on fish ecology, and it could be that cephalopod ecology has another trophic ecology as well. But yeah, and the other thing was that we had different distinct patterns for each single region. With the Mediterranean Sea with narrower trophic niche. That can be due to different sampling season. As I said, in the Mediterranean Sea we sample in summer with pelagic trawl. So it could be that we only have a subpopulation of the individual with only juveniles. And they didn't have time to display vertical migration, for example. But we can see that they have distinct pattern in each single region. And to emphasize on that, I did stable isotope trajectories analysis. So you can see the same for each region. The Bay of Biscay, the Gulf of Cadiz, and the Mediterranean Sea in the isotopic space, the trajectory across its life stages. So if you focus on the Mediterranean Sea, for example, the pink track is one individual. So the first one would be the core layer, so the born event. Then you will have some intermediate stage, and where you have the arrow is the end. So it's why working on islands is really interesting, because you can see how this isotopic trajectory across its life stages. And then the color represents different clusters. And as I said, we expected an increase of the nitrogen and the carbon, which we found, but really moderated for the nitrogen and for the carbon too. So they don't shift the trophic position in my results. And then we can see that each color is regionally, at the exception of the Gulf of Cadiz and the Bay of Biscay that share a common cluster. But I have to put some limitation here because I didn't have a lot of individuals for the Gulf of Cadiz. Where, for example, in the Mediterranean Sea, you really have a homogeneous cluster, that is the blue one, with only one outlier, compared to the Bay of Biscay that is really display really regional disparities with the gray cluster really lower or even a trophic position. So with that, I wanted to know where they were from, to know if it was really regionally within the Bay of Biscay. And within this map, you can see clearly that the color represent region, and that apparently the gray cluster represent the Asturia region. And I have a colleague that find also this, so it could be another population, but I will have to do some further analysis to confirm that. But there's a regional trophic plasticity for Ilex garnituti. And my last results will be the comparison between stable isotope signatures between mussel and islands. So here you have in A the nitrogen and in B the carbon, and for each single region, so the Bay of Biscay, the Gulf of Cadiz, and the Mediterranean Sea, with the estimated size and estimated age. And what is important is that the stable isotope signature of mussel is higher for each single region in nitrogen and carbon, compared to islands, that is the blue box plot, with each box plot represents one layer. So the first one would be the core layer, intermediate, and then outermost. For that, I need to say that in the previous animation, I kind of lie. The outermost layer is not really the time of capture exactly. You have another layer around it that is gelatinous, and this is a forming layer that for methodologic limits, we didn't put that layer in the analysis. So we kind of have a time window mismatch between mussel isotopic signatures and islands isotopic signatures. And this was to emphasize the importance of using multi-tissue analysis to have an overview of its diet. So that's the key takeaway that I want you to take. So first, it's really important of using multi-tissue analysis in trophic ecology. So that's why I use mussel and islands to have the overview. So around the life stages is important, but we also have to compare on other tissue. And then I found that Ilex garnituti displayed a trophic plasticity across the different Iberian waters, but also along the Bay of Biscay. And then I need to put some limitations. So I want to talk about the seasonality. Obviously, it's 3 big regions, so we could not sample at exactly the same time. So I know that isn't a limitation. It's why I discuss about it a lot. And then also the sample size in the Gulf of Cadiz, because we use pelagic trawls, so opportunistic fishing. So it's not that easy to catch Ilex garnituti actually. So it's a limitation, but for example, in the Mediterranean Sea and the Bay of Biscay, I had a good sample size. So some perspective that I will continue to do after in the EU of Santander, I will compare the diet with another squid. So Loligo with Galis. So I will do the same analysis and trying to compare to see if I see the same pattern. And then I want to integrate some metabarcoding to it for the subpopulation in Asturia cluster and even maybe in the whole different cluster in the Bay of Biscay. And also I want to continue the stomach content analysis that I started as preliminary results. So yeah, that's my perspective for the next month. And so thank you very much. Let's get it as go. I don't know if any is asked here. And yeah, I presented this at the Isobay at the International Symposium in San Sebastian in early June. So thank you.
Thank you. Perfectly on time. So, is there anyone that wants to start discussing? Nico?
Sure. Thank you very much.
In the order.
Well, thank you very much for this presentation, Lola. I really enjoy that. Although I miss more images of your nice squids there, no?
Yeah.
A little bit less text and more pictures of the squids, no? You really wanted to see the animal or more pictures on that. It would have been good. As being myself Asturian, I have to ask for. So I have to tell you that I'm sorry that this cluster from Asturias is so different.
No, it was actually cool.
Can you go back, please, to the map?
Yeah.
Where you.
Good.
Yeah. So your samples in the Bay of Biscay come from the Cantabrian Sea along a huge gradient, and it's much warmer at the coast of Basque Country than the coast of Lugo to the west. So I wonder if these environmental variables are important. What do you think of this?
So yeah, I thought about this. For example, also I want to emphasize also about the upwelling in Galicia. It could also be a source. The thing is, like on the paper, the upwelling over here will like more south of Galicia, and that when we are our sample are not like the effect of the upwelling is not that strong anymore on the paper. It could be, but it wasn't so.
But you mean at the moment of sampling?
Yeah.
But I mean, probably what you observe is the accumulated effect of the upwelling that happened before, no?
And yeah, that could be. But also, we also thought about, for example, the isotopic baseline. That could be different because of this upwelling, but we actually found, like, one colleague of me find that no increased gradient around the coast or decreasing gradient of the isotopic baseline in nitrogen. So yeah, we need to continue to investigate.
Yeah.
Because I cannot say that it's only the upwelling that
Okay.
explain these results.
Okay, it's very interesting. What about the depth? At which depth were these animals caught in the nest?
It was a pelagic troll, and so that's also, for example, like I said, that we have the pelagic troll during summer in the Mediterranean Sea. And for example, in other studies, they use like benthic troll, and they have a bigger individual, so it could be like the adults go deeper, while the juveniles are more pelagic.
And you know the exact depths of these pelagic trolls?
No, not exactly.
Because maybe, there could be a different. Do you know the depth preferences of these animals as they grow?
The things they do vertical migration in the day, but also around their life. But normally juvenile are more pelagic and they go deeper. But also during the day, they normally display vertical migration too. So they're really moving a lot. So it's why I said that it was hard to catch them, because it's they're not just on the substrate, for example. So yeah.
Okay, thank you very much.
Hi Lola. Very nice presentation. Thanks for showing us, even if not many physical squids, a lot of results. I really enjoyed. Can you maybe go to the before? So I really want to. Yeah, you showed a lot of graphs that had a lot of information. So I'm glad that you selected focusing maybe on some clusters. I'm not showing all the data that you have, and I'm sure that is a lot. So I thank you for that. So this is in the eyes, right? Or
yeah.
Okay.
Yeah.
So you were talking about seasonality, and this is of course influencing, but this would be somehow the reflection of all the life. So seasonality is already like included, or like variability according to seasonality is also within this data. So I think that you can really trust your data. The matter is finding an explanation, right? So for instance, have you figured out what they feed on? Like, are they different behaviors into different, like, well, I wouldn't say colonies, but regional differences in what they feed off? Because maybe here they have more like small fish, while in Cadiz or in the Mediterranean they have big fish. Or do you think that that's playing a role?
So that's why I start stomach content analysis. But for squid, I have a little problem is that they crush the prey. So in the stomach, I actually have some piece of things, but so I have some extra slides.
Nice.
So that was the things that I could use. Maybe it's a mix, a mandible, of crustacean. So we found that like in majority in whole the stomach. The problem was like nobody really did a book about mandibulas and which species exactly species is about. So it's really hard to know from that. So we kind of take. I had, for example, in one stomach a big piece of a head of an individual that we couldn't identify. So we took this individual from a frozen individual from other sample to compare the mandible, the big, from the one we were in the stomach. But then we tried with other individual from the same genera or other species, and they look really the same. And there's different morphotype. So for now, I cannot say, but other things that we found, like it's just fish eyes, so I can say that they're fish, but not more. And the more interesting will like statolith, but it's the same, it's not abundant. So yeah, we continue with that, but for now it's hard to say exactly what they eat.
Yeah, maybe with some genetics, but I know that's expensive, so yeah, I don't know. Maybe last question. So regarding the muscles, there are some like strange tendencies, right? So I would expect maybe more, of course not linear, but you see some changing patterns. So first I want to ask, what kind of adjustment did you use for this model? And besides, which is the interpretation that you give to these S-shaped equations? Well, correlations.
So I didn't put the muscle results here, but I did some analysis before, and it's so that for doing the analysis specifically on muscles, I take into account autocorrelation with coordinates, residual, and it was explaining a lot of this fluctuation. So it's just autocorrelation.
Because what would be expected? Like,
yeah, we expected it kind of smoother, but I guess it's science. Like for example, the Gulf of Cadiz is really, but as I said, I have a sample limitation too. So it could be also the sample size, because in the Mediterranean Sea is where I have most of my data, and it's smoother. So
more connected.
Yeah.
Okay, so you have still a lot to do with it.
Yeah.
Very good luck with that. Thank you.
Thank you so much. So I have here some curiosities. For instance, the number of individuals you come and see could not be so high, especially in Cadiz, because it was for not properly fishing, and not so high or good sampling. You don't prefer numbers. What number of animals? And
so for islands, I had only 10 in the Gulf of Cadiz, and then from of islands, but then for peeling the layer and sending to the lab and everything, I could have 3, so the core layer, the intermediate, and the outermost layer with enough data, with only 3 or 4. So
yes, that were the limitations. And in the other cases, in other sites that you consider a good number, what was the range of numbers that you would have for individuals?
So that in the other region?
In other regions.
So in the Gulf of Cadiz had 48 for islands and around 30 for the Bay of Biscay. So really, yeah.
Then my curiosity was here in these graphs for muscle and eyeglasses. You did not explain so much this axis of the X. What are the 3 groups of the high lenses?
So it's each layer So the first is the core layer,
Oh, it's
then
the layers.
intermediate, and then
Yes,
outermost.
yes.
Yeah.
Because below I see estimated size and
Yeah.
estimated age, and I was confused.
Yeah.
And then the muscle, that's why there's only one line, because it's the wall muscle.
It's
Yes.
just here.
And the muscle, I'm curious, which part of the squid?
It was the dorsal mental part.
Mhm,
Yeah.
mhm. And to finalize, not to take too much time, about the stomach contents, you are still analyzing. Any prediction of will it work well or not?
So
compared to
I
other
will work.
tissues?
The thing is right now is fundings. I need to find a contract money to continue.
Mhm.
So yeah, it's a bit uncertain.
Mhm.
But I will try to publish at least what I have right now.
Yes.
And
Anyway, in terms of tissues, for now, you will bet in the island. That will be the one that work better.
yeah, it's. I mean, it's just show different things.
Mhm.
It's not that it works better. It's just that it show a longer time period. So it's for me more interesting. But it's still, like I said, like it's still important to have the muscles and to use multi-tissue. There's some people that use beaks, for example, and not eyelenses.
So the better will be complementary tissues, but the highlights can look back in maybe in history.
Exactly,
and this
across the
information.
life stages.
Thank you so much.
Thank you, Lola, for very nice clear presentation and for introducing us to lenses way for stabilized of analysis. Very, very nice. I want to continue with that actually because I was also wondering, it's very nice with the methodology that you see the two different things based on the type of sample that you use. But I was thinking like why would eye lens be preferable in a way that you see the population, let's say the overall signature of the population or the overall traffic needs or whatsoever. But with the muscle you do see a short-term, like the closer past, let's say, of the animals. What are the questions that the two different methodologies answer, basically, that you choose depending on your question? Like, is it, for example, if we're trying to see environmental changes for trophic, responsive with trophic ecology, would that be more suitable to use which of the two, or what is your opinion?
Well, in a long-term change, I will say eyelenses because it can show from the birth to the adulthood if it changed or not. But I still think that different time periods are still important. I'm doing stomach content analysis and it's really a snapshot, but it's high resolution and even if it's time consuming and everything, it's still important to have all of this information to do a snapshot and kind of a patchwork of everything.
Also related with your sampling, I'm not very sure if I understood. Did you identify also like the sex of the animals or the age you try to estimate, I guess?
So the sex was hard because for the Mediterranean Sea, for example, there were a lot that were too little, so it was undeterminated sex. So I didn't include it for this reason because in one location I cannot use. There's like 2-3 individuals that has the sex determined, and here the estimated age is actually really to take with carefulness because it's just from literature that uses satellite to put an estimated age with the size. We cannot use that like that, but there's a PhD student that works on that to actually estimate the age with eye lenses. So that we'll see how it works.
That's quite interesting indeed to see. Also, you mentioned in the beginning in your hypothesis that you would expect an increase in delta nitrogen and carbon 13 with size and age. I understand this for nitrogen isotope, but I was wondering why would you expect this for carbon?
Because in literature they say that adults go more deeper because of physiological capacities. They can go in deeper water, so more benthic habitat. So it's why I expected a higher carbon.
Okay. And the last one, maybe if I may, I will just maybe not exactly a question, but a comment. On your tourism result, like interesting result about the contracted niche for larger size individuals, that you are wondering how to relate it or so. I was wondering, well, I'm not sure. I was just thinking if that can indicate a specialization that is related also with the age and the maturity of the animals.
Yeah,
I don't know the literature about this, but
like
just thinking.
initially I put specialization of the diet, and then I had a meeting with autotrophic ecology, and they said that it's not that easy to say specialization of the diet. So I changed to just contraction. I need more analysis to say it, but it looks like it. Yeah.
It's quite promising.
Yeah.
Thank you.
Thank you. Thank you very much, Lola. And we will now close this session. I would like to thank all the jury members and our three speakers, Elena, Marta, and Lola. Thank you very much.